video we discuss about the topic glycolysis and KREB cycle. That is very important topic in the biochemistry metabolism. Okay. First of all, you see this map. What this map tell about? Actually, this map contain all the metabolic pathway that was occur in our body in biochemistry. That all metabolic pathway are present in this diagram that called metabolic metro map. All metabolic pathway are connected with each other, and that all are present here. See, this is carbon fixation, this is pentose phosphate pathway, fatty acid synthesis, fatty acid elongation, beta oxidation, citric acid cycle. This is glycolysis. This process is the glycolysis, and this opposite of glycolysis is gluconeogenesis. And this glycolysis pyruvate, that is the product of glycolysis, is connected with the citric acid cycle with the help of acetyl coenzyme A. And this, with the help of this citric acid cycle, the urea cycle is connected, right? And with this urea cycle, amino acid deamination will occur. See here, here is amino acid deamination process. Okay. By this citric acid cycle, gluconeogenesis is connected with this glyoxylate cycle. So all metabolic pathway are connected with each other. The product or subproduct of the process. That subproduct are connected with each another type of cycle, like fatty acid synthesis. See here, the citrate of the citric acid cycle is connected with the fatty acid synthesis, right? And here this is connected to beta oxidation, acetyl coenzyme A. Okay. And again, the product of the subproduct of beta oxidation is connected with waxes, sphingolipid. So all the metabolic pathway that occur in our body or in other organisms, all are connected with each other, and that all pathway are start with which substrate? All metabolic pathway are started with which substrate? That is glucose. What? That is glucose, fructose, like the glucose or fructose. All metabolic process are started with the glucose or fructose. That is glycolysis. So starting of the all metabolic pathway is the glycolysis. That occur where? Glycolysis occurred where? Cytoplasm. Right? So glycolysis is the cytoplasmic pathway because they occur in a cytoplasm. That's why it is called as a cytoplasmic pathway. What? Cytoplasmic pathway. Glycolysis is the cytoplasmic pathway which break down glucose into 3 carbon compound and generate energy. What do the glycolysis? In glycolysis, 6 carbon compound, that is glucose, is break down into 3 carbon compound, that is pyruvate, right? And this process is known as glycolysis. This process is known as glycolysis. So, come, what is a glycolysis? Actually, glycolysis is the first stage in a cellular respiration. Already I told, glycolysis is the first step. Means it's starting of the all metabolic pathway. Starting of the all metabolic pathway is the glycolysis. This is the cytoplasm, and here the process of glycolysis will be takes place. Understand? So Greek glycolysis. Glycolysis is made up of what? Glycose and lysis. Glycose and lysis. Glycose means sugar, sweet in nature, right? And lysis means breakdown or dissolution. So, what do you mean by glycolysis? Means breakdown of sugar. What? Glycose means sweet, sugar, and lysis means breakdown. Then what happened? Breakdown of the sugar. Breakdown of sugar is known as glycolysis. So, glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. For cellular metabolism, that energy are in the form of ATP. What? ATP and NADP. Nucleotide adenine diphosphate. Right? This is the ATP and this is the NADP. That is the form of energy. Glycolysis consists of energy requiring phase followed by energy releasing phase. Actually, in glycolysis, there are two phases will be occur. In first phase, energy are used. Means ATP are used in first step. And in second step, that is called, first step is known as energy requiring phase. Energy requiring phase, because they utilize the ATP. They use the ATP. That's why that phase is known as energy requiring phase. And next phase is the energy releasing phase. What? Energy releasing phase. In this phase, ATP is released and NADH is released. Okay? ATP and NADH is released here. So, this stage is known as energy releasing stage. So, overall production of energy in glycolysis is 2 ATP and NADH. With the help of one NADH, with the help of oxidative phosphorylation, this NADH is converted into ATP. By which process? Oxidative phosphorylation. One NADH, with the help of one NADH, 2.5 ATP will be formed. What? 2.5 ATP. So, in glycolysis, the overall production of ATP is 2 ATP and 2 NADH. Then you convert that and get the total. Okay? This is the process. Glucose. This is a glucose 6-carbon molecule in cytoplasm. They converted into pyruvic acid plus energy. That is 3-carbon molecule. This is pyruvic acid. That is 3-carbon molecule. Another process. This process is known as glycolysis. Right? This process is known as glycolysis. In next process, this glucose molecule is converted into ethanol. That is 2-carbon. That is 2-carbon molecule plus CO2 and plus energy. This process not required oxygen. In the absence of oxygen, and in this process they require oxygen. So this is known as fermentation. What? Fermentation. This process is known as fermentation that occur in a yeast. Another is a lack of oxygen molecule in cell. They converted into a lactic acid. That is 3-carbon. 3-carbon plus energy. This process is occur in a muscles. Right? So these are the three process by which glucose molecule can be break down. First process is the glycolysis. First process is glycolysis. First process is glycolysis that occur in presence of oxygen. Second is fermentation. Fermentation that occur in the absence of oxygen and converted into ethanol. And third is lactic acid formation. Lactic acid formation that also occur in the absence of oxygen and form product that is known as lactic acid. So these are the 3 steps by which glucose molecule can be break down. Here we study of the process of glycolysis. Okay, here we study the glycolysis. Actually, in all 3 process, energy will be released, either in the form of 1 ATP or 2 ATP or 3 ATP, but energy will be released in all process. Here, glycolysis is the series of reaction that extract energy from glucose by splitting it into 3 carbon molecule that is called as pyruvate. Sequence of reaction converting glycogen to pyruvate, lactate or ethanol with the production of ATP as energy. ATP as the energy form. Reversal of glycolysis along with the alternate arrangement of the irreversible steps will result in the synthesis of glucose, that is known as gluconeogenesis. Gluconeogenesis is nothing but it is a reversible process of glycolysis. Reversible process of glycolysis is known as gluconeogenesis. Reversible process of glycolysis known as gluconeogenesis, that is from pyruvate to glucose. Pyruvate to glucose is known as gluconeogenesis. Okay, clear here? This is the steps of glycolysis and energy regeneration. This is glucose molecule. After breaking of glucose molecule, 2 pyruvate molecule will be generated. Remember this, because this is a 6 carbon molecule and pyruvate is the 3 carbon molecule. That's why total 6 carbon molecule, then 2 pyruvate will be generated. This pyruvate molecule is converted into acetaldehyde, after that ethanol. What? After that ethanol. Then this process is known as fermentation. This step is known as fermentation. In organism that perform cellular respiration, glycolysis is the first stage of this process. In every organism, breakdown of glucose is the first step of the metabolism. Because we eat anything that will be in the form of glucose. We take everything in the form of glucose. So every reaction will takes place in our body or in other organism body after the breakdown of glucose, either in the presence of oxygen or either in the absence of oxygen. But there is a breakdown of glucose will be happen. However, glycolysis doesn't require oxygen, and many anaerobic organisms that do not use oxygen also have this pathway. Glycolysis takes place in the cytosol of a cell, and it can be broken down into three main phases. Glycolysis takes place in the cytosol, right? And it can be broken down into three main phases. That is, first energy requiring phase, second is splitting phase, third is energy releasing phase. First is energy requiring phase, second is splitting phase, where actually splitting phase is not main, it is not important, but energy requiring phase and energy releasing phase is important. Splitting phase is the one step where glyceraldehyde 3-phosphate, 2 molecule of glyceraldehyde 3-phosphate will be formed. That is one step in the glycolysis. That's why it is not important. You remember only energy releasing phase and energy requiring phase. Glycolysis is the first step in a cellular respiration. Remember this. Here is a pathway of glycolysis. Glucose molecule is break down into 6. Actually, one thing that several enzymes are used. What? Several types of enzyme. Several type of enzyme are used here. Like hexokinase, phosphoglucosisomerase, phosphofructokinase, aldolase, glyceraldehyde 3-phosphate dehydrogenase. Remember this. If NADH, formation of NADH will be takes place, then dehydrogenase enzyme are used. For formation of NADH, dehydrogenase enzyme are used, and the formation of ATP kinase are used. Remember this thing. For NADH, dehydrogenase enzyme are used, and for ATP, kinase enzyme are used. These are important. So, first step, glucose. Glucose is converted into glucose 6-phosphate. During this conversion, glucose to glucose 6-phosphate, 1 molecule of ATP is required. What? 1 molecule of ATP is required here. This is a glucose molecule. This glucose molecule is converted into glucose 6-phosphate. 1, 2, 3, 4, 5, 6. So here 1 phosphate group is attached. During the attachment of 1 phosphate group, 1 ATP is required here. What? 1 ATP is required here. Understand? Next step is conversion of glucose 6-phosphate to fructose 6-phosphate. Glucose 6-phosphate to fructose 6-phosphate. So here what happened? Here is isomerization. Nothing will change. Only isomer will be changed. Right? Glucose 6-phosphate to fructose 6-phosphate. Here the 6 molecule cycle is present. Here 5 carbon cycle. Here 6 carbon cycle. Here 5 carbon cycle. So here isomerase enzyme is used. That is phosphoglucosisomerase. And here there is no requirement of ATP, and there is not a release of ATP. Okay. Next step is the conversion of fructose 6-phosphate to fructose 1,6-biphosphate. Means there is addition of 1 phosphate molecule on another carbon. Means during the addition of 1 phosphate, remember during the addition of 1 phosphate molecule, 1 ATP is required. In the first step, In the first step, at the 6th position phosphate group is added, then 1 ATP is required. Now at first position 1 phosphate molecule is added, means 1 ATP is required here, right? So here fructose 1,6 biphosphate will be formed. After that, this step is known as energy. This step is required energy, so this is known as energy requiring step. Energy requiring step. Because in this step, at two time, first and third, 2 ATP is required. Now this is known as, after fructose 1,6 phosphate, this is known as splitting phase. Here fructose 1,6 phosphate is splitted into 3 carbon molecule, that is glyceraldehyde 3 phosphate. That is glyceraldehyde 3 phosphate. See, 1, 2, 3. This is 3 carbon molecule. So this step is a splitting step. After this glyceraldehyde 3 phosphate, another molecule is formed, that is 1,3-bisphosphoglycerate. And remember, in the splitting phase, there are 2 molecules. Remember this, 2 molecule of glyceraldehyde 3 phosphate is formed. 2 molecule of glyceraldehyde 3 phosphate is formed. So after the 5th step, all the process will take at the 2 times, because there 2 molecule, 3 carbon 2 molecules are present, right? So each and every step will take at the 2 times. After the formation of glyceraldehyde 3 phosphate, this glyceraldehyde 3 phosphate is converted into 1,3-bisphosphoglycerate. 2 molecule of 1,3-bisphosphoglycerate. During the formation of this, one molecule of NADH is released. Remember this. One molecule of NADH is released. Already told, during the formation of NADH, which enzyme is used? Dehydrogenase. During the formation of NADH, dehydrogenase enzyme is used. And there is 2 molecule of glyceraldehyde, means 2 in 1 NADH molecule is multiplied by 2, means 2 NADH is used, is formed. Not used, it is 2 molecule of NADH is formed, right? After 1,3-bisphosphoglycerate, this 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Means there is a conversion or transferring of phosphate group will be takes place here, right? So during the transfer of the phosphate group, 1 ATP is formed. During the addition of phosphate group, ATP is used, right? And during the removing of the phosphate group, 1 ATP is formed. Remember this. During addition of the phosphate group, energy will be required, and during the removal of the phosphate group, energy will be released. Remember this is. So 1,3-bisphosphoglycerate will convert it into 3-phosphoglycerate. That is for this formation will takes by which enzyme? kinase, because here is a removal of one phosphate molecule, and during the removal of the one phosphate molecule, one molecule of ATP is formed here. And we remember that here is 2 molecule of 1,3-bisphosphoglycerate is present. So 2 ATP is formed here. After the formation of 2-phosphoglycerate, it is converted into phosphoglycerate, 2-phosphoglycerate. Here only mutarotation will takes place, and it is the enzyme that is used is mutase. Another is a enolase enzyme is used, and phosphoenol pyruvate will be formed. During the formation of phosphoenol pyruvate, they also contain phosphate group. So we want to remove all phosphate group. So during the removal of the phosphate group and formation of pyruvate, one ATP molecule will be released. And the enzyme that is used here is a pyruvate kinase. Okay, understand? Clear this? This is all 10 steps. In glycolysis, total 10 steps will be there. Okay. How much energy will be formed here? See here. ATP used, that is 2 ATP, right? 2 molecule of ATP will be used, that is -2. ATP that is formed is here 2 ATP will be formed, and here 2 ATP will be formed. Total 4 ATP plus 4 ATP. And NADH, 1 molecule of NADH will be formed in each step, means 2 NADH molecule will be formed. Two molecule of NADH will be formed, and with the help of one NADH, how much ATP will be formed? 2.5, means with the help of 2 NADH, 5 ATP will form. Then how much total ATP energy will be released in glycolysis? 5 plus 4, 9, 9 minus 2, 7. 7 molecule of ATP overall. 7 molecule of ATP will be formed in glycolysis, right? Understand? It is clear? Clear this is? So next is here energy requiring phase. Already understood what happened in energy requiring phase? This is 1 molecule of glucose, and in energy requiring phase, this 1 molecule of glucose is converted into 2-glyceraldehyde 3-phosphate, and during the formation of this 2-glyceraldehyde 3-phosphate, 2 ATP will be required, right? During the addition of phosphate group at the 1 and 6 position of the carbon. 2 ATP will be required. So this process is known as energy requiring reaction. What? This process is known as energy requiring reaction. In this phase, the starting molecule of glucose gets rearranged and 2 phosphate group, remember 2 phosphate group are attached to it. The phosphate group make the modified sugar, now called as fructose 1,6-bisphosphate, that is unstable, allowing it to split it in half and form 2 phosphate bearing 3 carbon sugar. Because the phosphate used in this step come from ATP, 2 ATP molecule get used here, right? Already I told you that during the addition of phosphate group at 1 carbon position and 6 carbon position, 2 ATP molecule are used, right? Clear? So this process is known as energy requiring phase. Next step is splitting step. Splitting step means where fructose 1,6-bisphosphate or diphosphate is dissociated into glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. And this dihydroxyacetone phosphate is again converted into glyceraldehyde 3-phosphate. This process is known as splitting step. Another is an energy releasing phase. In energy releasing phase, glycerol with the help of glyceraldehyde 3-phosphate converted into 2 molecule of glyceraldehyde 3-phosphate converted into 2 pyruvate, that is 3 carbon compound. During the conversion of the glyceraldehyde 3-phosphate to 2 pyruvate, 2 molecule of NADH and 4 molecule of ATP will be formed, because phosphate molecule are removing. 2 phosphate molecule are added in earlier, 2 phosphate molecule are added, right? So now we want to remove that phosphate molecule for stability. When you removing this phosphate group, ATP will be released. So there is two molecule of glyceraldehyde 3-phosphate will be formed. Means with the help of 1 molecule of glyceraldehyde, 2 ATP will be formed. So 2 molecule means 4 ATP will be formed, right? So this process is known as energy releasing reaction. In this phase, each 3 carbon sugar is converted into another 3 carbon molecule, that is pyruvate, through a series of reaction. In this reaction, 2 ATP molecule and 1 NADH molecule are made, because this phase takes place twice, because 2 molecule of glyceraldehyde 3-phosphate will be formed. Once for each of the 3 carbon sugar, it makes 4 ATP and 2 NADH overall. Understand? This step is clear? This is the overall glycolysis that convert 1,6 carbon molecule of glucose into 2,3 carbon molecule of pyruvate. The net products of this process are 2 molecule of ATP and 2 molecule of NADH, right? See, this is actually in metabolic reaction there are 3 stages. That is, first stage is glycolysis. Second step is conversion of pyruvate to acetyl coenzyme, and third step is TCA cycle, that is tricarboxylic acid cycle. So in all steps the energy will be released. Overall energy will be released in this all step. So now we study about the first step. Here we study about first step, that is glycolysis. In glycolysis, overall energy is 2 ATP and 3 ATP, net yield 2, because 2 ATP is already utilized in first stage, that is energy requiring stage, right? So here is 2 ATP, and with the help of NADH 3 ATP will be formed, but actual from 1 NADH 2.5 ATP will be formed. Means with the help of 2 NADH 5 ATP will be formed. Okay, clear? How many ATP? 5 ATP. So overall energy in glycolysis is 7 ATP. This is the step. That is, glucose is converted into glucose 6-phosphate with the help of, these are the steps that is in detail. These are the glucose is converted into glucose 6-phosphate with the help of hexokinase. Glucose 6-phosphate is converted into fructose 6-phosphate with the help of phosphoglucose kinase, and fructose 6-phosphate is converted into fructose 1,6-bisphosphate, means addition of 1 phosphate molecule with the help of kinase molecule, And this step is the splitting step. What? This is the splitting step. Splitting step means the fructose 1,6-phosphate molecule is splitted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and again dihydroxyphosphate is converted into glyceraldehyde 3-phosphate. Okay. In where ATP is added means step where phosphate is added means ATP is utilized. Remember this. And kinase enzyme are used. Okay. Understand? This is the energy requiring phase. This are a step where which enzyme is used, which are isomer, like that only. Already I told you this all steps. First step where glucose is converted to glucose 6-phosphate, 1 phosphate molecule is added, 1 ATP is utilized here. In second step, glucose 6-phosphate is converted into the isomer, that is fructose 6-phosphate. In third step, another phosphate group is added at one position of the carbon of the fructose, right? And 1 ATP is utilized here, right? In fourth step, fructose 1,6-bisphosphate is split into two molecules, that is DHAP and glyceraldehyde 3-phosphate. And in fifth step, DHAP is converted into glyceraldehyde 3-phosphate because it is a stable product. DHAP is not stable. That's why it's converted into glyceraldehyde 3-phosphate. Another this step is a energy releasing step. In this step, glyceraldehyde 2 molecule of glyceraldehyde 3-phosphate that is formed is converted into 1,3-bisphosphoglycerate, and here dehydrogenase enzyme is used and NADH will be formed. Means 1 molecule of NADH will be released here. 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Here 1 phosphate molecule will be removed. 1 phosphate molecule will be removed here. This phosphate molecule or this phosphate molecule will be removed here. Means 1 ATP will be released, and in this process 1 phosphate molecule will be removed. In this is removed. Here also 1 ATP will be formed and converted into pyruvate molecule. So these are steps are the energy releasing step. Already 6, 7, 8, 9 steps are you already know. 2 half reaction occur simultaneously, means glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Then after that 1,3-phosphoglycerate is converted into 3-phosphoglycerate, right? 1 ATP will be formed here. In above step 1 NADH is formed and dehydrogenase enzyme are used there. In 8th step, mutation will be takes place or isomer will be formed, that is 2-phosphoglycerate. In 9th step, 2-phosphoglycerate loses 1 molecule of water molecule and form a phosphoenol pyruvate, that is PEP. In last step, removal of the phosphate group will be takes place, removal of phosphate group and releasing of 1 ATP molecule, and that converted into a stable product, that is a pyruvate. Pyruvate is the end of the glycolysis. Actually, this you recall all the step of the glycolysis. During the recalling, there are inhibitor are also present that inhibit the process of the glycolysis. Not at every step of the glycolysis inhibitor is the present. There are several steps where inhibitor are go and bind with that enzyme that convert the substrate into product and inhibit the all process, like arsenate, iodoacetate, arsenate and iodoacetate. They inhibit the arsenate and iodoacetate inhibit the glyceraldehyde 3-phosphate dehydrogenase enzyme. Inhibit the glyceraldehyde 3-phosphate dehydrogenase enzyme. Another is bromohydroxyacetone phosphate. They inhibit the dihydroxyacetone phosphate. That is formed during at the splitting stage, right? During the inhibition of formation, during they inhibit the dihydroxyacetone phosphate, means one molecule of pyruvate will be formed only. Because during this splitting of the fructose 1,6 bisphosphate, two molecules will form. One is glyceraldehyde 3-phosphate and another is dihydroxyacetone. And after that, this dihydroxyacetone will be converted into glyceraldehyde 3-phosphate. When bromohydroxyacetone inhibit this dihydroxyacetone phosphate, they not converted into glyceraldehyde 6-phosphate. Means one molecule one step will be inhibited. Means one molecule of pyruvate will be formed only, not two molecules of pyruvate. Another is fluoride. Fluoride inhibit enolase. Enolase are used where? Where removal of one molecule of water will be takes place, means at the 9th position. When they inhibit the PEP, formation of PEP enzyme, if PEP molecule will be not formed, then pyruvate will be not formed. Means the inhibition of total 2 pyruvate molecule. Means 2 pyruvate molecule will be not formed. Because fluoride inhibit the enolase. That is 9th step of the glycolysis. 9th step. Another is oxamate. They inhibit the lactate dehydrogenase. Actually, lactate dehydrogenase are not used in this glycolysis. They used during the formation of lactic acid. During the formation of lactic acid. Not in this 10th step. In 10th step, iodoacetate, bromohydroxyacetone and fluoride. These are the main inhibitor in the glycolysis. Right? In glycolysis, arsenate iodoacetate inhibit the glyceraldehyde 3-phosphate. If glyceraldehyde 3-phosphate dehydrogenase will be not formed, they inhibit the formation of glyceraldehyde 3-phosphate dehydrogenase enzyme. Means there is no formation of the pyruvate molecule will be takes place. Or another step after that, this is the 6th step. 6th step, right? 6th step of the glycolysis. After 6th step, rest 4 step will be not takes place, right? This is the step where inhibitor are present and inhibit the process of glycolysis. Here is this red see, this is a pyruvate kinase. This is a phosphofructokinase. Okay. So here glucose 6-phosphate inhibit the hexokinase 1. This is the overall inhibitor after TCA cycle also. The overall inhibitor that inhibit the glycolysis process. The ATP citrate that form that will inhibit phosphofructokinase like that. But we study that in earlier slide, in previous slide, we studied that enolase. This enolase is inhibited by fluoride. Okay, means this step will be not takes place after inhibition, right? Iodoacetate inhibit the phosphodehydrogenase enzyme, right? At this step, this is inhibited by iodoacetate, right? Means this process will be stopped. Bromide will be inhibit the DHAP. So these are the inhibitory product. This all about the process of glycolysis. Now come with at the TCA cycle. What is TCA cycle? TCA is the tricarboxylic acid. What? Tricarboxylic acid cycle. TCA is located in a mitochondrial matrix in eukaryotes. In prokaryotes, it's located already in a cytoplasm. What is the main goal of the TCA cycle? The main goal of TCA cycle is the breakdown of pyruvate into a CO2 and acetyl coenzyme A and release more energy. What happened here? Break down the pyruvate or pyruvic acid into a CO2 plus energy, so that more energy will be formed. Next process, what is the process? Each pyruvate loses one carbon and make a 2 carbon molecule, that is acetyl coenzyme. Actually, before TCA cycle, one step is there where pyruvate is converted into a acetyl coenzyme, because pyruvate is a 3 carbon molecule. Pyruvate is a 3 carbon molecule. They in TCA cycle require 2 carbon molecules, so they converted into a 2 carbon molecule, that is acetyl coenzyme A. The carbon join with the oxygen that breathe in to create the carbon dioxide we exhale. The acetyl coenzyme can then diffuse into the matrix of the mitochondria. This acetyl coenzyme may go into a mitochondrial matrix, and where TCA cycle will be takes place, right? This is the TCA cycle. Here what happened? This is a pyruvate, that is the product of the glycolysis. This pyruvate is a 3 carbon molecule converted into a 2 carbon molecule, that is acetyl coenzyme A. This acetyl coenzyme A go and in TCA cycle in the mitochondrial matrix and combine with citrate, combine with oxaloacetate and form a citrate. This citrate is 1, 2, 3, 4, 5, 6 carbon molecule. Then is converted into cis-aconitase. Cis-aconitase converted into diisocitrate, then alpha-ketoglutarate, then succinyl coenzyme A, then succinate, fumarate, malate, oxaloacetate. This all step will be continuously takes place after the addition of acetyl coenzyme A. So during the formation of this process, during the process of this cycle, citric acid cycle, here NADH, FADH and GTP or ATP will be formed here. So more energy will be released here. What? More energy will be released here. Here see, during the conversion of diisocitrate to alpha ketoglutarate, one molecule of NADH will be released. During the conversion of succinyl coenzyme A to succinate, one ATP or GTP will be formed. One GTP will be formed here, right? And during the formation of fumarate to malate, one FADH will be released. And during the malate to oxaloacetate, one NADH will be released. So overall production of the NADH FADH is the 3 NADH, 1 ATP and 1 FADH will be formed here. So huge amount of energy will be released here. This NADH, the NADH that are formed in glycolysis or TCA cycle, this all NADH and FADH go into a oxidative phosphorylation. For the formation of ATP. Right? This FADH, NADH go into a process where oxidative phosphorylation, that process where ATP will be formed from this NADH and FADH, right? These are the process of the TCA cycle. Now, since there are two molecules of acid, remember that two molecule of pyruvate will be formed, means two molecule of pyruvate will be formed, then two molecule of pyruvate will be converted into two molecule of acetyl coenzyme A, and two molecule of acetyl coenzyme A will enter into TCA cycle. Means TCA cycle will takes place at a KREB cycle, or TCA cycle takes place at 2 times. What? 2 times. So, there must be 2 turn of the cycle will takes place, one for each pyruvate. This is the one cycle by one pyruvate. This is the another cycle by another pyruvate. Okay, clear? Next, what is the reactant and what is the product? Product from glycolysis. What is the product of the glycolysis? 4 carbon molecule will be recycled. What is the product of acetyl coenzyme A? ATP. Sorry. Here, what is the product of glycolysis? The product of glycolysis is the pyruvate. Okay. Product of acetyl coenzyme is the 4 carbon molecule that is recycled. Product of ADP is ATP, and product of oxygen is carbon dioxide. FAD is FADH2, and NAD plus is NADH. Okay. So these all are the product. What is the reactant? What is the product? What happened? Next is inhibitors of inhibitor in citric acid cycle. The inhibitor of citric acid cycle is fumarase. What? Inhibitor of citric acid cycle is malonate, arsenate, and fluoroacetate. Fluoroacetate inhibit the aconitase. Arsenate inhibit the alpha ketoglutarate dehydrogenase, and malonate inhibit the succinate dehydrogenase. Fumarase is an enzyme, fumarase is not inhibitor. Fumarase convert the fumarate into an L-malate. So there are three inhibitor in the citric acid cycle. First is fluoroacetate, second is arsenate, and third is malonate. Okay. Next is significance of TCA cycle. TCA cycle are a complex oxidation of acetyl coenzyme. They are help in a generation of huge amount of energy, that is form ATP, NADH, or FADH. Final common oxidative pathway, integration of major metabolic pathway. Fat is burned on the way of carbohydrate. Excess carbohydrate are converted as neutral fat, and no net synthesis of carbohydrate from fat. Carbon skeleton of amino acid finally enter the TCA cycle. These all are the significance of the TCA cycle. So, here we stop here. Now, all TCA cycle and glycolysis will be completed.